Hello all, my name is Dave Sandmeyer. I'm teaching the Bio 345 AP&P2, General Principles of Human Anatomy, Physiology, and Pathophysiology this spring. It's one of my favorite courses. I hope you like it as well. I will be posting these YouTube lectures, and typically each of these recorded videos, there'll be a link to them in the course Blackboard site as described in the email. And during these video periods, I will refer to PowerPoint slides that are also available on your course Blackboard site. Whenever I do that, I'll put a yellow tab with a slide number at the bottom like this, which says slide 1. That means that if you open up your Unit 1 PowerPoint presentation and go to slide 1, that's what we're talking about. I will also integrate those slides into the video before posting it on YouTube. So this is Bio 345. Most of you have just taken Bio 245. I want to, before we get into too many details regarding the syllabus, I want to tell you about a few quick changes based upon your feedback from last fall. First change, while the class sessions will still be pre-recorded and made available on YouTube, I'm also going to have, for every day's lecture, that night, from 8:30 to 9:30 p.m., an open Zoom session, Q&A session for that day's lecture. This will allow you guys to get more feedback from me regarding that day's lecture. It'll allow you in sort of a Q&A, questions and answers session, hear other students' questions. I think that'll be really valuable as a learning resource. So be aware of that. I will also record all of those Q&A sessions and post them on YouTube as well. So if you can't get to one of them, you'll still be able to benefit from hearing from other students' questions. Of course, you're always welcome to visit me during either my office hours or outside of office hours by Zoom as well. And if you want to do that, just send me an email with your blocks of free time, and I'm happy to schedule you in. The more we interact outside of class, I think the better you guys will do in this course. It's a really fun course. The human body is amazing. I hope you'll appreciate that as we go through the various systems in this next semester. Another change from last semester, the essay exam will be changed to a multiple choice exam. More details on that later, but it'll allow me to ask you more questions to cover the breadth of the material rather than just 8 questions as we did on the material last time. In addition, again, based on your good feedback, I will make available a study guide for each unit. If you look in the Unit 1 folder of the course content on Blackboard, you'll already see there's a Unit 1 study guide in there. That is the bare bones sort of outline of what, in the end, you should focus on for these exams. Also, prior to the exams, as always, I'll be giving review sessions independent of those 3-time-a-week Q&A sessions that I mentioned earlier. And I will distribute some sample multiple choice questions prior to the exam so you guys can get an idea of the sort of the level of questioning I might use for those exams. So those are some changes. Also, I've asked Dr. Grimm for the lab part of the course to now post his worksheet grades for you guys. I understand those weren't posted last semester, so those will be posted. In addition, based on your feedback, the course syllabus, which I'm going to show you here in a minute, has consolidated all due dates. So if you look at the course syllabus carefully, even the lab due dates for various lab assignments are listed in the lab schedule near the end of this syllabus. So I hope these changes improve the course, and I hope they make it a more enjoyable course for you guys to get through and learn from. Now, I'm not going to go over all the details of the course syllabus, but if you look at slide 1, that's page 1 of the course syllabus. As always, I give an overview description of the course at the top with some policies on the bottom half of page 1 regarding COVID and other expectations. If you move along to slide number 2 in your PowerPoint presentation, that is page 2 of the course syllabus. It gives you my name, my office hours, Dr. Grimm, his office hours, how to contact us, and the schedule for the course. Also, the various lab section schedules are shown there, and you've already been sent a student cohort list for this semester. At the bottom there, the textbook is the same one we used for the first semester. I strongly encourage you guys to dig into the online resources that go along with that textbook. They're super valuable. All sorts of tutorials, videos, animations, unlabeled pictures, labeled pictures, etc. Really, you should use that to the hilt this semester as a really good way to piece your way through the physiology. The lab manual is the same as last semester. We'll be using it more extensively in your lab setting. If you go to slide 3 of the course presentation, that's page 3 of the course syllabus, outlining some of the course objectives and student learning outcomes for this course, a list of the systems we're going to be doing. So we're going to learn all about the endocrine system, the cardiovascular system, the blood, immune system, respiratory, urinary, digestive system, and some highlights from the reproductive system. So hopefully we'll start to see how all of these organ systems piece together to form a working whole organism called a human. If you look at slide 4, that's just the next page. The grading system is illustrated there, 5 equally weighted exams worth roughly 2/3 or 65% of the course. And then again, you'll be having case studies and lab practicals or quizzes and other lab assignments which make up the remainder of the course. The grading scale in the middle of that page is the one set by our college. I have no control over it. So that's the one we are instructed to use. And then at the bottom of page 4 begins a day-by-day description of what we're going to be talking about. So for the first day there, January 20th, you can see it right above it says Unit 1, Endocrine System, and it gives the reading assignment in the textbook for the entire endocrine system. And then if you page through the rest of the syllabus, or the rest of these slides on your course presentation, you'll see that that day-by-day schedule continues all the way through with dates noted when exams are due, or when exams will occur for all of the 5 units of the course. And then on page 9, which is slide 9 of your PowerPoint presentation, you'll see the lab schedule, at least the first part of it, with the lab number and date on the left column. There's 2 dates there because there's lab sections on both Monday and Tuesday, so whichever one is relevant to you, obviously, is the one you're paying attention to. And then a description of what's going on in each lab. I've also highlighted the quizzes or lab practical dates, the case history due dates, and the due dates for all the worksheets or other lab assignments. So all of that is on one schedule, one syllabus for your reference. Okay, so let's start with the endocrine system. That's the first system we're doing in this semester, the endocrine system. And of course, the endocrine system is that system of glands that produce hormones that regulate activities in the body. It is a regulatory system, just like the nervous system that we learned about last semester is a regulatory system. In fact, the endocrine system and the nervous system are the 2 great controlling systems in the body. They are what control most of the physiology, how the body works. So if you take a look at slide number 15 in your PowerPoint, that slide shows an individual with highlighted a lot of the different endocrine glands. You can see they're spread throughout the body. And these endocrine glands, so basically the endocrine system is a system of endocrine glands. Endocrine glands. First of all, let's break this word down. Endo and crine. Crine means to secrete something, to release something from a cell. And endo means within. So the endocrine system is a system of glands that secrete hormones within or into the bloodstream. So, as I said earlier, those hormones are regulatory chemicals. They help control activity in the body. And so the endocrine system is, along with the nervous system, the other great regulatory system of the body. So So it's the other great regulatory system. Helps control stuff. And we'll see lots of examples of that as we move along in this unit. So if you take a look at slide number 15, you see all sorts of different endocrine glands. You see the thymus, the thyroid, the parathyroid, adrenal glands, pancreas, etc. These are different from exocrine glands. So these are endocrine glands. Remember, exocrine glands are those glands that secrete substances through tubes that ultimately lead out of the body. Whereas the endocrine system, it's those glands that aren't associated with tubes or ducts. In fact, the endocrine system consists of the ductless glands. Ductless glands. In other words, unlike exocrine glands such as sweat glands, mammary glands, etc., these ductless glands, they secrete their substances directly into the bloodstream. So these ductless glands secrete hormones directly into the bloodstream. No, there's no duct system involved here. All right, so these hormones are regulatory chemicals. So let's just make that real clear here. Hormones are regulatory chemicals. By that I mean they can bind to receptors on target tissues and regulate what happens to those target tissues. All right? So they work by binding to receptors, specific binding to receptors, in target tissues. Okay? By target tissues, I mean tissues and other structures, organs, etc. in the body that are influenced by, affected by the binding of these hormones to those receptors. Now, when I say specific binding, what I mean by that is each hormone chemical is of a specific size and shape. So it won't just bind to any old receptor. There are specific receptors made of protein that have a specifically shaped and sized binding sites. So hormones they don't just randomly bind to anything, but they bind to specific receptors by a hand and glove sort of mechanism. So the way this works is you might have an endocrine gland consisting of a bunch of cells. I'll just draw a clump of cells here like so, each with a nucleus inside it. And let's say those are some endocrine cells. The glands have a very good blood supply. So here's a blood vessel coming in, maybe a capillary. And what would happen is that those endocrine gland cells will release hormones. These little green dots are hormones into the bloodstream. And those hormones will get whisked away through the bloodstream to other far reaches of the body. And in some distant place in the body, maybe over here somewhere, there may be a target tissue, a bunch of cells, part of a tissue of an organ or something like that. And those target tissues might have receptors on their surface for those hormones. So what happens is the hormones get dumped indiscriminately into the bloodstream and circulate wherever blood circulates. But those hormones might be able to jump back out of the blood and through the interstitial space and bind to receptors in target tissues. And that sets into motion certain effects. So over here, again, we have the endocrine gland releasing a certain hormone. And over here, we have some target tissue that's going to respond to the effects of that hormone. Okay? So one important point about this is these hormones are chemicals. They're just molecules. They don't have a brain. They don't know where they're supposed to go in the bloodstream. They don't know where their target tissue is, but they're basically just getting whisked through the bloodstream to circulate through all parts of the body. And they might jump out of the bloodstream in all parts of the body. But the only place where those hormones will have any effect is where they can bind to a specific hormone receptor. So that's how hormones sort of know where to go. They don't really know where to go. It's just that they'll only bind because of their specific size and shape to very specific receptors. So if a target tissue doesn't have any receptors shaped like this, it won't be affected by this hormone. But if the target tissue has this shape of receptor, it will bind to that hormone and it will be affected by that hormone. And the binding there, again, of the hormone to its receptor will trigger a bunch of events inside the cell, oftentimes ultimately influencing what enzymes get made or how active certain enzymes are, which then determines which chemical reactions take place in those cells, which determines what sort of physiologic events happen in that cell. Now, compare this system, this endocrine system, to the other regulatory system. Remember, the nervous system. Remember how that worked. In the nervous system, the way that control was exerted was by neurons. So we drew a typical neuron. It might look like this, with the big long axon, has some branch points at the end of it, and then of course its synapses with other neurons, or muscle cells, or even gland cells. And the way this nervous system worked was that action potentials we learned about travel down axons, causes the dumping out of neurotransmitters that then bind to some receptors on the postsynaptic neuron here. Now, that's a pretty short trip from where the neurotransmitter gets released to where it binds to a receptor. And remember, the action potentials travel at pretty fast velocities as well. So the nervous system is set up for the most part, faster responses than the endocrine system. Because the endocrine system, the way it works is it has to dump a hormone into the bloodstream. That hormone has to circulate throughout the blood, jump back out of the bloodstream into the interstitial space, and then bind to some hormone receptor. That process takes longer than this process in the nervous system of a really lightning quick action potential causing release of neurotransmitter to bind to some receptor. So the point about this is that the nervous system is specialized for quicker responses, more rapid responses, whereas the endocrine system the responses are slower in onset, create more delayed responses compared to the nervous system. It's just because of these differing mechanisms. So the endocrine system creates more delayed responses. Also, I will say that the nervous system, the responses are shorter lasting responses. They don't last as long, in general. Whereas the endocrine system, even though the responses are more delayed in terms of when they occur or their onset, the endocrine system generally creates longer lasting responses. And we'll see why that's the case later in this unit. But longer lasting responses. Now, there are two chemical classes of hormones that I want to talk about. Two chemical classes of hormones. There are first the hormones that are amino acid based or amino acid derived. My shorthand for amino acid is always going to be a capital A, capital A. So amino acid based or amino acid derived hormones. So what I mean by that is the hormone might actually have been metabolically produced from a precursor amino acid, or the hormone itself might consist of a small number of amino acids linked together. All right? So generally speaking, this category of hormones, this chemical class of hormones, generally are water soluble. Usually, I'll say, there are some exceptions to that, usually water soluble hormones. And then the second class, chemical class of hormones, are the steroid-based hormones. The steroid-based hormones. So the steroid-based hormones are usually produced from cholesterol or other steroid, other types of steroids. So they're produced from precursor steroids, oftentimes cholesterol. Okay? And generally speaking, the steroid-based hormones, those are lipid soluble. Unlike the other class. So those are lipid soluble. They don't dissolve well in water. They're hydrophobic, whereas the other class of hormones are generally mostly hydrophilic. Okay, so some examples of each of these. First of all, examples of amino acid based or amino acid derived hormones. Well, they might be things like ADH, antidiuretic hormone. TSH we'll talk about, thyroid stimulating hormone. These are examples of hormones in this category. Insulin and glucagon. All of these hormones are actually polypeptides. So these are examples of polypeptides, chains of amino acids linked together to create a hormone. Now, the steroid-based hormones, examples, I'll just give a few here. I won't list all of them. But for example, cortisol, estrogen, testosterone, aldosterone. These are all examples of steroid-based hormones. Another important concept about these hormones is that sometimes hormones are sometimes released in an inactive or less active precursor form. They're sometimes released into the blood in either an inactive or less active form. And then what happens is that they then get chemically converted to the active form once they're at the target tissue. So they get converted to the active form of the hormone. In the target tissue. You might ask, well, why would this be the case? What's the purpose of this? Well, it's one more way to make sure that a hormone only has an effect in the appropriate target tissues and doesn't have effects elsewhere. So for instance, a hormone may only act on a certain tissue because that tissue has enzymes that are able to convert that hormone into its active version. Whereas other target tissues might not have the enzymes that convert that inactive hormone to an active hormone. All right, so this is one more level of control to make sure hormones only have effects where we want them to have effects, which is kind of interesting. The less active or inactive form, there's actually a name for that. It's called a prohormone. So if you see that term, prohormone, it means a less active or inactive form of a hormone. The other thing that's important to know about hormones is that they actually themselves don't last very long in the bloodstream. In other words, hormones have fairly short half-lives. Fairly short half-lives. In other words, they're broken down pretty quickly. And ultimately removed from the body, the breakdown products, removed from the body. Either typically via the bile or urine production. All right, so it's kind of interesting. These hormones have fairly short half-lives. Well, wait a minute. Didn't I say earlier that the effects of hormones are long-lasting, often longer-lasting than nervous system effects? And it's true. I said that. But the point of this is that the hormone itself just triggers the first step in a long cascade of responses in target tissues. And so the hormone itself just has to create a binding to a receptor, and that unleashes an avalanche of other activities that continue on long after the original hormone is gone from the system. So this actually doesn't contradict what I said earlier. Hormones have long-lasting effects, but the hormones themselves don't last very long. So they're removed. They're converted to metabolites that are inactive, either in the target tissue or in the kidney and the liver, and ultimately removed from the body. Okay, now, how potent will a hormone's effects be on a target tissue? Well, it turns out there are certain factors influencing this. I'll call these factors affecting hormone potency, how well they work, in target tissues. Okay? How strong an effect do these hormones have? Well, it depends on a number of factors. First of all, as you might expect, the concentration of the hormone in the bloodstream would matter, obviously. So concentration of the hormone in the blood. Obviously, if there's more of that hormone in the bloodstream, that hormone's going to have a stronger effect. But that's not the only thing. Another factor that influences how potent a hormone is the hormone receptor affinity. Hormone receptor affinity for binding. What do we mean by that? What we mean by that is how strong a bond is it between the hormone and the receptor? Is it weakly binding and maybe get released too easily? Or is a stronger bond that hangs out and the hormone hangs out on that receptor for a longer period of time? It turns out that there's a degree of regulation of this affinity. It turns out that certain hormone receptors can be better receptors for hormones or worse receptors for hormones, oftentimes based on whether that hormone receptor is phosphorylated or not by, say, a protein kinase enzyme. So the concentration of a hormone matters, and how strong the bond is between the hormone and its receptor also matters. But there's a third effect that I want to mention as well, and that is the number of receptors in the target tissue for that hormone. The number, literally how many of them are there? The number of receptors in the target tissue for that hormone. That also matters. And that also is regulated to some degree. We can upregulate or downregulate the number of receptors for a particular hormone. Now, obviously, if there's more receptors on a target cell for a hormone, then the hormones are going to have greater effects. But if there's few to no receptors, there's not going to be as much of an effect. So let me give you an example of how this works. This number of receptors is regulated. So, and this is regulated for various purposes. We can upregulate or downregulate the number of receptors. Let me give you a couple of examples here. And the first example is one of upregulation of receptors. This is sometimes referred to as the priming effect. Priming a target tissue to be really super responsive to some hormone as it circulates through and binds to receptors. So this is upregulation. This typically happens in cases in which there's been chronically low levels of a particular hormone over a period of time. So due to chronically low levels of a hormone over time. All right? It's sort of the body's way of saying, hey, we haven't seen much of that hormone lately. We need that hormone's effects. Let's maximize the effects of the meager amounts of hormone in the bloodstream by increasing the number of receptors for that hormone. So even if the hormone itself remains at low levels in the blood, if a target tissue doubles the number of receptors for that hormone on its cells, then that can help upregulate the effect of that hormone, sort of priming that target tissue for that hormone's effects. That's why it's called the priming effect. Okay? The opposite of that would be downregulation, and this occurs in some cases as well. There can be downregulation of the number of receptors for a particular hormone. This is sometimes called desensitization. It's the opposite of the priming effect. Desensitization. And in this case, what would happen is if there's chronically high levels of a hormone over time, a target tissue might be getting overactivated by that hormone. So let's put the brakes on, so to speak, and let's at least reduce the number of receptors in that target tissue for that hormone so that that hormone's effects aren't too exaggerated. So downregulation would be due to not chronically low levels of a hormone, but chronically high levels of a hormone over time. To try to to try to minimize the exaggerated effects of a certain hormone. So be aware that a hormone's potency, how well it works, is influenced by all three of these factors. Okay, now let's get into the nuts and bolts of this and talk about the mechanism of action of hormones. How do these hormones actually create effects in target tissues? Okay, so the mechanisms of action of hormones. Now, remember earlier I drew this picture to show you guys that hormones are just sort of released into the bloodstream and they will whisk away to wherever blood goes in the body. So hormones will circulate through the entire body. But remember, we said the hormones will only have specific effects on target tissues that have receptors for those hormones. So that's one way to narrow down where a hormone might have some effect. But my question for you guys is, where are these receptors and what happens when a hormone binds to a receptor? So let's talk about that a little bit here. First of all, where are the receptors? Where could they be? So let's talk about that. Where are the receptors? And first of all, if this is a target tissue again with some cells, like so, like we drew earlier, and a neighboring blood vessel with hormones coming from the bloodstream into that area like we drew earlier. Okay, so here comes the hormones. One place where those receptors might be is in the nucleus of the cells. So the way this would work is that a hormone would come out of the bloodstream through the interstitial fluid, and if it's lipid soluble, pass easily through the phospholipid bilayer and get deep into the cell, deep into the nucleus of the cell, and bind to some receptor that's either next to or directly attached to some portion of DNA. So what would happen then is that binding of that hormone to its receptor and then the subsequent hormone receptor complex binding to DNA would influence which parts of your genome get transcribed and translated, and which proteins get made, for instance. So we generally see hormones have receptors in the nucleus of the cell, typically for lipid-based or lipid-soluble hormones. Okay? For lipid-soluble hormones. In other words, the hormones themselves, usually steroid-based, are lipid-soluble, so they can get through this cell membrane, this phospholipid bilayer, and get deep into the cell. But not all hormones are lipid-soluble. Some are water-soluble and can't easily move through cell membrane. So where's another place where receptors might be? The receptors might be right out on the surface, as I drew in that previous diagram. So for instance, you might have hormone receptors embedded in the cell membrane of the target tissue cell, with the binding area poked outward, sticking outward, so this hormone can come in and bind. Now, that is a place where you see receptors specifically for hormones that are water-soluble hormones. Okay? Because obviously hormones that are water-soluble, that don't dissolve well in lipids, they wouldn't even be able to get through the phospholipid bilayer cell membrane deep into the cell. So the hormones that are water-soluble, they sort of have to ring the doorbell on the outside of the house. They have to bind to some receptor on the outside of the cell to create internal activities through so-called second messenger systems. So these are the general locations where we see receptors for hormones. Now, sometimes hormone receptors are in the cytoplasm, and so a lipid-soluble hormone might get through the cell membrane, bind to a receptor in the cytoplasm, but ultimately, in those cases, the hormone and receptor complex makes its way into the nucleus and then binds somewhere on the DNA, again, to influence which parts of your genome get transcribed and translated. So we will stop here for this lecture segment, part 1, and we'll continue part 2. And when we do that, we're going to get into the actual mechanisms. What happens when a hormone binds, for instance, to a receptor in the cytoplasm or deep in the nucleus, versus what happens when a hormone binds to a receptor on the outside of the cell membrane. So we'll see you at the next lecture segment.